Genomics session. Our first speaker is James Zhou from Stanford University.
All right. Great. Well, hope you are still awake and energetic in the last day of the short program. Thanks again to the organizers for a really fun week. So I'll talk about some pretty recent work on what we're calling contrastive learning. And most of the work is actually really driven by two excellent graduate students at Stanford, Abu and Martin. So they're really terrific students. So, okay, is that better? All right. Okay, great. So yeah, so I'm at Stanford, and I'm very, very fortunate to work with very fantastic students at Stanford as a part of the machine learning and computational biology group there. So basically, half of my group is working on trying to really push state of the art in statistical machine learning to develop new algorithms and new foundations, and the other half takes these technologies that we develop and apply them to quite a wide range of biomedical applications, all the way from genomics to health. And I have two students here, so I want to highlight them so you can go find them during the breaks. So Jaime, so he's working on quite interesting problems of how do we combine statistical guarantees while you're still using powerful, for example, neural networks, right? So he has a paper that just came out recently on giving provable false discovery rate controls, so provable FDR controls, while you're using arbitrary general neural networks, right? So very interesting results. So go talk to him if you're interested. So Brian, he's a computer science PhD student. He's also working on quite a few interesting projects. One is on doing predictions on point clouds, which has a lot of interesting applications to single-cell RNA-Seq data, for example. He's also working on this problem that we're calling data sculpting, which is used optimization to try to construct training data sets, right? So if you don't learn anything else from the talk, the main takeaway message is you should go find them during breaks, and then they'll tell you more about their projects. Great. So the kinds of applications that I'm interested in my group is quite diverse. As I mentioned, so we're interested in everything all the way from the molecular side, where we have machine learning models for genome editing, for human cell atlas, all the way to much larger scales of, for example, using smartphones to track mosquitoes and to model EHR records of hundreds of millions of individuals, right? So the data that we look at comes from very different modalities, and they span also quite a wide range of physical scales. So in many of these data applications, what we find very useful as the first initial step of analysis is usually to do some sort of data exploration or some sort of unsupervised learning or dimensional reduction, right? So here's probably a step that's very familiar to many of you here. So if I give you a data set, right, usually one of the first things you do is to do some sort of visualization of that data set, right? Maybe you use PCA or you use t-SNE to look at your data, right? So so this is a particular real data set come from protein abundance. The details of data is not so important, but if you do your PCA on this data set, and if you see this plot, right? So maybe you'll be less excited, right? So there's not that much, seems like that much interesting structure in this data set. Right, but if you take the same data set, but if this is the plot that you see, then maybe you're more motivated, right? So then you see there are these interesting clusters here in data, and then it leads to natural hypothesis that you might want to try, right? You might want to ask, okay, so are these two clusters of points, are they different in some meaningful ways? And if you do that actually for this particular data set, you'll find that these two clusters are actually biologically meaningful, right? So if you had done this downstream follow-up experimental analysis, you find that, okay, so these gray dots correspond to sort of essentially healthy mice, and the red dots correspond to mice with Down syndrome, right? So they're interesting biological differences. But the point is that in order to even know what follow-up analysis to do, right, so it's quite important for your initial data exploration and data visualization to reveal to you what are the interesting structures in these data sets, right? And that's really one of the main goals of unsupervised learning. So what I want to show you today is that actually these two plots actually come from the exact same data set. It's the same data set being analyzed, but it's analyzed using two different methods. The first one is using the standard unsupervised learning methods, for example, PCA. And the second one is what I'm calling contrastive learning or contrastive PCA. right? So the same data set, but it just depends on how you analyze them, what models you use, you can actually really find very different patterns. And that could lead to very much different downstream analysis. And I'll come back and describe this in much more detail, but just to point out that if you actually do any of the standard unsupervised learning methods, dimensional reduction methods, on this data set, they would not have been able to find these kind of interesting patterns, right? So it is something quite crucial about doing the contrast here that's useful. So before I dive into the exact details of what contrastive learning is, so to give you more of a high-level motivation for what we're trying to do here. Right, so we developed this framework in a NIPS paper that I wrote a few years ago. And there's been sort of a large community of follow-ups, both by our group and by our colleagues, on this idea of contrastive learning. So what is contrastive learning? So the idea is that if you're given a data set, right, so the standard approach to do unsupervised learning is that you want to find patterns that are dominant in a particular data set. Right, so if you do PCA or autoencoders, linear or nonlinear methods, you're always trying to find what are the dominant patterns in one particular data set. And I claim that that's actually not really, in many applications, that's not really what you really care about. Right? In many biomedical settings, what you're really interested in is what are the patterns that are enriched in one data set in contrast or in comparison to some control data sets. Right? So I don't really care about just the dominant patterns in a particular data set. I care about modeling the contrast, right? So, and that's actually turns out to be quite useful as an approach. So what I want to show you today in a very simple example is how do we actually really think about doing this contrast and how do we model this? So everything I talk about today in the next few slides can be generalized to much more complex nonlinear models. I'll describe that more at the end. But I just want to really go through step by step a very simple case, which is how do we do a contrastive PCA, right? Which is already quite useful. And if you have any questions, you should feel free to ask, because otherwise then I'll ask you questions during the talk. So far, so good? Okay. Okay, so here's the simplest setup, right? How do we do a contrastive PCA? So the high-level idea here is that, so imagine if I have this toy example, I have two data sets. I have my green dots, that's what I'm calling my target or foreground data. That's my data set of interest. So here are the green dots, right? And if I do regular PCA on these green dots, this is the line that I will learn. This is the subspace that explains the dominant pattern, right? So it turns out that for this particular data set, if I take this green line here, and if I project onto that subspace, I would have not observed. I would have missed the interesting patterns, the interesting clusters, which are these two clusters, right? So in this, I have a second data set in this example, which corresponds to the blue dots. So the blue dots here, that's what I'm calling the background, right? So that's some sort of background or control that you want to do your contrast against. So the background will have some differences from my foreground, from my target data. But in this case, the background seems to share a lot of the common subspace, right? It also has a lot of variation along this diagonal subspace. So ideally, what I want to do in this case, when I do the contrast, is that if I want to find patterns that are the most enriched in my green dots compared to my blue dots, then I want to find this red subspace, right? But because that red subspace, that's the subspace that explains a lot of the interesting variation in my foreground data that's not shared with my background data, right? So intuitively, whatever contrastive learning methods that we want to do, we want it to be able to find this red subspace. Yeah.
Is it basically just picking out some lower order principal components and figure out which ones are the most important ones? And it seems like here it might be the second principal component.
So right. So in high dimensions, right? So it turns out that the red subspace and the green subspace I'm drawing here, they're not necessarily orthogonal, right? So there are settings, and I'll show examples of this, where does not exist a lower order any principal component on my foreground data that reveals interesting patterns. Good question. Yeah?
Can you please explain again what is the background data?
Yeah, so the background data in this toy example are simply the blue points, the blue dots. Right, so I'll explain in detail in a few slides of how exactly we select background data, but you should think of it as being, if I have some experiments, I usually have some controls. Right? So I'm interested in what are the patterns that are enriched in my experiment compared to my controls. I use my control data as my background. Okay, so I'll go through this very carefully, so we're all on the same page, right? So I have some data set. It's my target data set X. So each row of X corresponds to 1 point. It could be a high-dimensional data set, have some points. Okay, so the simplest case of, if we're talking about subspaces or PCA, essentially what we're doing is we're looking at different directions, and we're looking at variance explained by each of those subspaces, by those directions. Right? If I have a particular direction V, a particular vector V, what I'm doing is I'm going to project each of my data points onto V, right? And I'm interested in what is the variance of X that can be explained by V, right? And that's simply given by this formula here. Right? So it's very easy to parse this. Right? So X is my data set. X times V is the projection onto this direction. Right? And since I'm interested in variance, which is a square, I'm just going to basically square this. And that's my variance explained. Right? Okay. So I can do this for any direction in my data space. And I'm interested in finding which direction that explains the most variance, that corresponds to the most informative subspace. Right? If I do that, if I solve this optimization problem, I'm solving exactly the standard PCA. Right? That's the first eigenvector. So, okay, so that's the standard PCA approach. And again, all of this can be extended to nonlinear models, to kernel PCA or autoencoders, but I want to go through the simplest case. So now the setting I'm looking at for contrast learning is that I have my foreground data of interest, that's my experiments, and I have some background, that could be my controls, right? So it has a different set of samples. They could have different numbers, right? So, and for each direction, I'm interested in what is the variance, what is the information of that direction that that direction explains of my foreground data, right? So that's the first equation. Yeah, so that's this equation here. And then the second equation is how much of the same direction, the same subspace explains of my background data, right? So intuitively, my goal is to find subspaces that explains as much of my foreground as possible and as little as my background as possible. Okay, so let me actually go to the board so I can draw a better picture for you than I could on PowerPoint. Okay, so the idea here is that so I start with some high-dimensional data sets. So I have some high-dimensional space. I have two data sets, right? So I have that lives in that space. I have my green dots, it's my foreground, and I have my red dots, that's my background. Right, so they live on high-dimensional space. So intuitively, what we want to do is that I want to find, can people see this? Okay, so I want to find a projection onto some subspace V so that the picture looks like the following, right, after I do my projection. Right, so my foreground data is going to be, maybe they're interesting clusters. And my background is going to be somehow collapsed into some very small, near the origin, right? So ideally, if I can do this, if I can explain out of my foreground variation and very little of my background variation, then I found a good contrastive subspace. Okay, so let me explain what the bottom graph is. So let's call this. So I have many choices of what are the possible directions, what are possible subspaces to use, right? So that corresponds to I can pick any of my unit vectors that's in this d-dimensional space. V1, V2, right? So for each of these unit vectors, for each of the V's, I can compute these two numbers, right? The variance explained of V by V of my foreground data and the variance explained by V of my background data. So I get 2 numbers exactly for each direction. So then, so that's the variance explained. So then I can look at the following plot, which is variance explained. Variance explained of the foreground, variance explained of the background. Basically, I can project all of these vectors onto this 2-dimensional space. And for an interesting reason, that I don't have time to explain here, that actually they'll map this unit sphere here onto a convex region in this 2-dimensional space. V2. Okay. So now the question to you is, right, so if you look at this 2-dimensional region, what directions would you want to select as the most interesting contrastive directions? Any thoughts? Okay, so I just have this 2-dimensional region, right? So just tell me what part of this region looks the most relevant to you if you want to find the contrast. Okay, great. So faculties are not allowed to answer, but that's good. Okay, so right, I would want to find regions that are somehow down here, right? Why is that? Why do I want to find regions? Why are these the good subspaces, the good vectors? Right, because if I'm down here, then I explain the most of my foreground X, right? And that's why I'm going to the right. And since I'm in the bottom, I explain very little of my background data. Okay, so the second question is there one particular direction V on this boundary that you think is the best one? Or do you think that they're all quite good? Okay, yeah, good. So a priori, so it's not clear which one of these do I want to use. Right, so that people also call this the Pareto frontier, because all of these points cannot be strictly improved upon by any other directions. Okay, so our goal then, in some sense, is I want to find this entire Pareto frontier. I want to find this entire red curve that corresponds to the right bottom frontier of this whole region. Right, so that corresponds to potentially some complex set of subspaces in my original high-dimensional space. And I claim that we actually have an algorithm that finds exactly that Pareto frontier, and it's a very efficient algorithm. Right, so I want to find that contrastive subspace. So the theorem that we can prove in the paper is that if I want to find exactly that, this red boundary that corresponds to my optimal set of contrast, I simply just have to solve a sequence of problems, eigenvector problems, that looks very similar to my original PCA. Right, so that's the set of problems. So I have some contrast parameter alpha. Alpha is non-negative. And what this term corresponds to is the first term is basically the covariance of my foreground data. And I'm simply just going to subtract away some alpha times the covariance of my background data. Right, it's as simple as possible. Right, so it just corresponds to this the simple notion of contrast. And it turns out that if you actually vary alpha from 0 to increase it to be large, what you do is that you exactly span out this the most contrastive directions, right? This lower red boundary. Okay. So in this linear case, also I'll talk about generalizations in the nonlinear setting, but in this linear case, so to find the contrastive subspaces corresponds to exactly solving this simple modified eigenvector problem. Right. So that's what we're calling contrastive PCA. Questions about this? In the back. Good question. Yeah, so alpha equals 0 that I wrote on the plot is exactly the regular PCA. That's right. As I increase alpha from being larger than 0, I get something different from a regular PCA. Yeah.
You're trying to make sense of your background data.
Okay.
That, kind of a thinking is kind of a mixture of your the structured data.
Yeah. Okay. So let me explain that now. Right. So great. So that's a good segue. So so I told you what the algorithm is, what the mathematical properties you want to satisfy, but there's still several questions that are that we need to consider. Yeah, one more. Turns out that that's not an issue. So this doesn't have to be positive. So let me actually show you. So I'll come back to that in a bit. Right. But there are three main discussion points, things that we need to consider. Is first, to answer the nice question, so how do we actually think about how do we choose background data? Right. The second is how do I actually choose this contrast strength alpha? Right. And then how do we generalize this to nonlinear models, for example, to autoencoders? So I'll spend most of my time talking about the first two. So how do I choose background data sets? Right. So in most practical setting of interest, the data set that you analyze, you usually have some natural background that you want to compare against. Right. So oftentimes if you run an experiment, you have some control group or baseline group, and that could be your natural background. Right. Or if you have a time series data, you have a later time point 1, maybe you can use your previous time point 0 as the background. Or you can use do your post-treatment contrast against your pre-treatment. The point is that there's not, so you do have to exercise your own best judgment, your domain expertise in choosing what the background data set is. So I'll show you 3 examples of contrastive PCA applied to different data sets and to different backgrounds. So the first one comes from application in population genetics. So here, each dot is one individual. We have genotypes of these individuals. So here, the individuals come from Mexico. And many of you have seen these kind of plots of regular PCA applied to genetics data, right? So you often want to see something where the individuals, if you plot them on the regular PCAs, their position on that PCA captures or corresponds to geographic position. So in this case, I have an interesting data set. That's actually from our collaborators, Carlos Busmonte's group. So we have individuals from 5 different states, 5 different locations in Mexico that I color-coded in different colors here. The colors of the states map onto the colors of the dots of the individuals. So if I apply the regular PCA, which is the first plot here, there's really no interesting differences between the individuals, right, that I can show from my PCA. It certainly does not map onto the geography. So here I'm going to use contrastive PCA with the automatically chosen contrast parameter, and it is able to much better find the geographic patterns in this data set. So what is the background here? So the reason why if I just apply the regular PCA to this data set, it's not able to find the interesting geography here is that for each of these Mexican individuals, you have a mixture of ancestry from Native American ancestry and European ancestry. Right. So because if I just take those individuals, their genotypes by itself, right? So the dominant patterns are going to be dominated by variations between Native American ancestry and European ancestry. And if I apply PCA to it, it's going to capture that direction of variation. Right? It's not going to capture the fine-grained variation in the Native American ancestry within Mexico, right, in different geography. So that motivates a very natural choice of a contrast. So I simply just take other individuals from Dowson genome with European ancestry and Mexican ancestry, right? So I just take that a publicly available set of that as my background. So we just plug that into the contrast PCA, and that's how we generate the second plot. Right. So the general principle I want to illustrate here is that I first have to think about what is the dominant pattern in my data sets that's salient to me, right, so that I'm interested in, and then I want to try to find a background data set that essentially I can contrast to remove the dominant patterns. Yeah?
Yeah, I just have a question, since this is a concrete example. So in this case, I mean, one very naive thing you could do is to just PCA the Europeans on their own and take the orthogonal complement direction to that PCA, and then, basically remove that the European direction from the Mexican population, so that you're not looking at that when you PCA the remaining data that you're interested in.
Yeah.
Why is
That's a great question. So if I just simply take the European subspace and look at the orthogonal complement of that, so it turns out that that would correspond to a particular choice of alpha, which I'll show in a bit is actually not as good as the choice of alpha that we find through this contrast, right? So the general framework that I wrote down here, so it does not assume that the subspaces, the contrasted subspace, necessarily has to be orthogonal to my background subspace, right? And it turns out that there are settings, including this one, where not being orthogonal is actually a better choice.
I'm confused by that. Sorry, not stuff, but why would that be the case? Because I assume that Europeans don't have Native American DNA from Mexico, so why would it not be the orthogonal?
Yeah, so maybe let me go through a few more slides and we can discuss that more offline, because I'll show another example of this where I think that'll become clear. Great. So, okay, so I'll try to go through this more quickly, so I have a few, just in the interest of time. So here we can also apply it to single-cell RNA-Seq data. Right, so each dot now is one cell, so you have a high-dimensional gene expression vector for each cell. So the left part is my regular data set, right? So I have cells from different individuals from pre and post treatment, right? So the contrast here is that I would like to. So the contrast. Okay, so the contrast here is that the background I use is basically cells just from one healthy individual, right? The idea being that I would like to, whatever the cell-to-cell heterogeneity, the variation in my healthy individual, that's the variation that I'm less interested in. So I would like to contrast it away so I can focus on the interesting differences pre and post treatment. So the third example from a very different setting, so you have mobile health data. So here you basically track different sensors of an individual. So here you have individuals doing two kinds of activities. The red dots corresponds to individuals who are cycling, and the blue dots corresponds to individuals who are doing some squatting activities, right? Very different activities. But if you just look at the regular PCA, right, of all these dots, they're not able to distinguish between these two activities, right? Because again, there's a lot of variation in the foreground data just across the different individuals, across the different sensors. So for contrast here, I'm just going to, again, I want to remove, contrast away those kind of variation. So I'm going to use my background, just a set of individuals who are lying still, right? So they're not doing any strong physical activities, right? So that's going to find the variation just between the different sensors. And again, if I use that contrast with different automatically chosen set of parameters, we can very clearly distinguish between the two populations. So the main thing I want to take away here is I want to use this contrasted PCA and contrast learning in general mostly as a tool for visualization, for data exploration, right? So this is not the final word on this. So you still have to go in and do your downstream analysis to each of these subgroups. But at least this makes it very easy to visualize what are the interesting patterns that are enriched. OK, so the second point I want to, yeah?
So in the regular PCA, you're not adding the background data at all, right? That one doesn't have the background feature matrix.
That's right. OK, so the second question that I promised to tell you is how do we actually automatically choose the contrast alpha? Right, so here's, I think, an interesting plot to illustrate that. So the x-axis of the plot corresponds to different values of alpha from all the way from 0 to large, right, in log scale. And for the y-axis, what I'm plotting is basically the angle of that subspace. Right, so what does that mean? So when alpha equals to 0, as I said, so that corresponds to just the principal components, right? So that's the regular PC subspace. And as I increase alpha, I'm going to start rotate that subspace when I do the contrasted PCA. Right, so at first, so the y-axis corresponds to how far am I rotating that subspace. Right, so you see quite an interesting transition phenomenon. So when I first rotate that subspace, I'm going to, when I first increase alpha, we're rotating that subspace fairly slowly. And then you see these sharp jumps, right, where you essentially move on to very different subspaces. And there's an interesting geometric reason why that happens. But that actually makes it very useful because then in order just to show a few values of the contrast that's the most interesting and representative, essentially I just want to show the values of alpha and the plot of that at a few different subspaces, at a few different clusters. Right, so basically what we do is that we just simply spectral cluster the different subspaces and then pick one representative subspace from each of those spectral clusters. So that's how we automatically choose alpha. So here I'm showing you an example where you have different values of alpha, starting from alpha to 0 on a synthetic data set. Right? So the synthetic data set, I've built it so it has 4 clusters in a fairly high dimensional space. The 4 clusters correspond to the 4 colors. So the regular PCA, because the data is in quite high dimensional space, it's not able to really disentangle these 4 clusters. Right? And this angle plot corresponds to exactly this data set. So if I start to increase the values of alpha, then I find essentially these 3 interesting regimes where we can find different subspaces where it's very clear what these patterns are. And it turns out that in this case, for this data set, the way it's constructed is it's actually information theoretically impossible for non-contrastive methods to be able to identify the interesting clusters. It's just because without doing contrast, the signal-to-noise ratio is too low. And this just illustrates that if you apply any of the standard dimensional reduction methods, linear or nonlinear, you can't really disentangle these 4 patterns. Right? So here the contrast is actually really useful to find the most salient patterns. Yes?
What are you using for contrast here?
Great. So the contrast here is just one of the clusters. Right. For background. Okay, so there are several extensions of contrastive learning. So we can do contrastive learning with kernels. Right? So it's one very simple way to make it nonlinear. So something that we're working on now is actually how to do contrastive learning with autoencoders. So the idea here is that, right, so we don't have to do contrast in just the original feature space. I can use a neural network, whatever your favorite representation is, to find a better representation of my data and then do a contrast in that representation space. The third direction is how do I actually do contrastive learning with more general latent variable models, right? Basically, I want to fit a generative model to the data set. So just one quick example of the third point, how do we do contrast learning with generative models? Right, so here's what we're calling contrastive topic models. So the idea here is that if I have a regular data set, I can fit a topic model or a latent Dirichlet allocation LDA model. So we had a talk earlier about these LDA models. So the input into the data set would just be you have a large corpus of documents. In this case, I have some documents from a particular set of US newspaper articles. Right? And here the columns here corresponds to the top 5 topics that are inferred by running vanilla LDA on this corpus. Right? So you find a lot of basically finance-related, financial market-related topics. So here, just as a proof of concept, right, I want to take as my background some other set of newspapers that also has a lot of finance, right? Something like maybe like Wall Street Journal. So I have another set of documents that has economic information that's my background, and I do my contrast. So the contrastive topic model in this case would learn the bottom set of topics, which are actually very different, right? These are the topics that it thinks are very different between these 2 sets of newspaper articles. And you see that it learns very different topics, including like golf, hockey, basketball. I think this one must be baseball, right? So by doing the contrast, you can really find interesting salient features that are different between the foreground and background corpus. So I won't have time to go into this, but the main idea of how do you do the contrast here is really through the method of moments, right? So the picture I was drawing before, essentially what we're doing is I have some moments, in this case, the covariance matrix of my foreground data, I have some moments of my background data. For contrastive PCA, the simplest case is I'm simply just subtracting away the background moments from the foreground moments. Right, so in general, I have some more general ways to modify, to basically project away the moments of my background from the moments of my foreground data. That's essentially what the general contrast is trying to do. And once we have these contrastive moments, then we can guarantee that we can learn the contrastive latent variable models. Right, so that's essentially what the method is trying to do. So just to wrap up, so we have a little bit more time for discussion. The high-level message I want to convey here is that in many cases, the standard unsupervised learning method is really not capturing what we would like to capture, right? If I'm interested in really the enriched patterns, which is often the case in biomedical data analysis, then I really want to essentially do something like contrastive learning or unsupervised learning was compared to a particular background data. And we've seen that this could be useful for both for dimensional reduction and also for visualization. And intuitively, the reason why this helps is that we would like to concentrate our modeling resources, right? We'd like to concentrate the capacity of our models. If I'm fitting my model to just one data set, the modeling capacity is going to be dominated by the dominant patterns in that one data set. But if I know that I'm interested in particular kinds of salient patterns, enriched patterns, then by doing the contrast, I can better focus the modeling capacity of my generative model onto that particular enriched patterns, right? That's really the motivation, the key idea behind the contrast. And in some settings that we can construct, we can actually show that contrast analysis is able to find patterns that are just information theoretically not possible to be identified using uncontrastive dimensional reduction methods. And again, so here are the fantastic students in my group who really led a lot of the works and ideas here, and sort of an obligatory picture of the nice California landscape in the background. I'll take more questions. Thanks. Yes? Good question. Yeah, so in the application we're looking at so far, the background has to be the same dimension as the foreground. The number of data points could be very different, but it should live in the same feature space. Yes?
What was the background data, for example?
Yeah, so there the background is just from a control box.
In your experiment, did you test for different size of the background data set? Because, I mean, if you have a few data points from background data set, then maybe the result will be different. And my second question is that, I'm to say that this method works when we assume that the variation of background data set is greater than variation of arbitraries, because otherwise the PCA capture what you want.
Yeah, okay, two good questions. So for the background data set, we do need to have this. Yes, so if there's too few points, then the contrast is not robust. So in the paper, so we have some statistical analysis using standard sort of concentration inequalities of what is the rate of convergence of the contrast dimensions, and it's essentially the same as the rate of convergence of regular PCA. Right? So basically, if you think that you need 100 points for regular PCA to work, then you should have around 100 points for the contrast PCA to work. So the second question is, yes, how much variation, how much signal should be in the background? Right? So the algorithm is actually very robust to that. So basically, we've done experiments where for example, we could synthetically reduce the amount of signal in the background. So that just corresponds to making the algorithm learn a larger contrast parameter alpha. Right? So there's a natural scaling between alpha, the size of the alpha, and how much signal there is in the background. Yes?
What happens when the alpha parameter...
Sorry, so I think there's another question here.
You wanted to prove that it's contrastive PCA, but you already knew that the groups are separate. And what would be the discovery example?
Yeah, these examples really try to show what are the different usage scenarios of contrastive PCA, right? And as I mentioned earlier, I would view the contrastive PCA as an initial step in your analysis, right? So if you really want to make and validate your discoveries, then you do have to do the follow-ups. So it's basically similar to how you would normally use t-SNE or PCA, right? So think of that. Then in those sort of settings where you should also think about how to use contrastive PCA. It's basically a way to do visualization. So the discoveries needs to be validated as you do with the regular PCA. Yes?
What happens when the contrast parameter alpha is big enough for the difference of the matrices to no longer be positive definite?
Yeah, so basically, right, so here we can increase the alpha to be very large, so on log scales. So the alpha here we look at very large. So as alpha becomes large, then essentially more and more of the eigen subspace will start to have negative eigenvalues, right? And when alpha becomes too large, then you're also basically finding uninteresting subspaces. Why? Because when alpha becomes too large, you're essentially, I think, doing what Lior is saying. You're essentially finding subspaces that have to be completely orthogonal to the information in your background, right? So that could just also be dominated by noise. Yes?
Thank you for this wonderful talk. So is that true that for any two like arbitrary matrices like X and W, can we always find an alpha that separates them with this contrast?
Well, so here X and W are not arbitrary matrices, they are covariance matrices, right?
Without any two arbitrary covariance. Like you can always get a covariance with that.
Yeah, so if I can always take, so that corresponds to I can always take some background data set, I can always do the contrast. If I just do that arbitrarily, I'm not going to find any interesting patterns, right? So I still have to pick my back.
Technical question instead of application-wise. So I'm
Yeah.
just wondering the nature of this.
Yeah, so the algorithm runs if you just put in any two data sets, right? With any two covariance matrices, the algorithm runs. But it's not going to give meaningful results.
Maybe later.
Okay, yeah.
Let's thank the speaker one more time.